el doctor Michael Chancellor, que es el director de Neurología del Departamento de Urología del Hospital William Beaumont en Royal Oak, en Michigan, Estados Unidos. Nos va a hablar específicamente de medicina regenerativa para la incontinencia urinaria de esfuerzo y vejiga hipocontráctil. Dr. Chancellor, thank you very much for being with us. And let's get started. Thank you.
Good morning. It's a topic I'm very excited about because I really believe it's the future of urology and all of medicines, regenerative medicine, using stem cell to modulate a dysfunction and to restore normal function. And just to introduce where I am, if you see on the map here, and I got the mouse pointer. Can you see my mouse pointer? Can you see my mouse on the computer? Okay, yes. So the picture on the bottom, that's Beaumont Hospital. It's one of the biggest, busiest hospitals in the US, and it's located here in Detroit, which is the automobile center of the United States. And that's where the clinical trial Beaumont is. We have a new medical school about 8 years old. But a little bit down and in blue is Pittsburgh. This is where I am right now. And this is where the company Cook Mouse site is and where the cell therapy is being manufactured. It really has one of the most advanced, sophisticated, and quality control stem cell manufacture facility in the country, in the world. I think the only bad part is we're both in very cold climate and we're both frozen right now. And this is where I am right now in this building, Cook Mouse site. It's a FDA and the European EMDA certified stem cell manufacture facility. So the cells can be shipped here, manufacture, and send back for injection anywhere in the world. And this is the people that's at Cook Mouse site. The person on the right, they're the vice president of the company. This is at a meeting at a little bit warmer location. And Ryan, a few years ago, came down with me when we visited Bogotá at Mauricio. And Ron Jankowski is in charge of the clinical trial. And this was us a few years ago at Mauricio's outside of the building. So I started off with the idea that stress incontinence is important, and we have good surgery, sling, bulking agent, and yet the real problem is a weak sphincter muscle. Is it possible, rather than stitching it up or filling it up, can we actually make the muscle better, regenerate the deficient sphincter? And the second problem that we've been attacking is the detrusor underactivity or the underactive bladder. When the bladder is failing due to muscle or innervation, and it's a very common problem with aging, with diabetes, is it possible to strengthen the weakened detrusor muscle to make it contract better? Underactive bladder. So to begin with the end in mind, research said we want to achieve a treatment that's simple, safe, and potentially effective. Can we do a simple biopsy to access the stem cell from a patient, himself, herself, this autologous muscle-derived cell, expand it out, pure, adequate number, and then inject it back into the sphincter or the bladder, but doing so as an outpatient, avoiding surgery, and that's the goal. Since the bladder and sphincter are muscle, my interest is instead of using hematological stem cell, embryonic stem cell, and fat stem cell, is can we biopsy the stem cell from the patient's own muscle? Because really, if you think about it, the skeletal muscle has the most plasticity of any organ in our body. What other organ can be as slender and run 26 miles in a marathon or be Arnold Schwarzenegger? Just with exercise. And skeletal muscle almost never have cancer. So it's a very safe source of cell therapy. And here's a picture. We did analysis, histology of the urethra, and we find that the sphincter is predominant striated, but the internal sphincter is mostly smooth muscle. So this is the area we want to target. When we started this, now it's been it's about 20 years now, we realized that we first had a proof of concept, but there was no animal model to demonstrate that it works or not. So before we can try it in human, we first had to create a stress incontinence animal model. And this is to show you, we took a pediatric Foley catheter, we distended the rat's vagina to create a birth trauma stress incontinence, and we can put in a multi-lumen 3 French transducer to measure the urethra and leak point pressure. So here's a rat. The balloon demonstrates the vaginal distension, creating a stress incontinence. And this is a suprapubic tube with the transducer. We're infusing methylene blue. And to make the rat leak, we take one of the whiskers from the rat and we tickle the rat's nose to make it sneeze. And then we look for, right here, leakage of methylene blue as the leak point pressure. And this is one of our earlier experiments that we showed that control, this is the leak point pressure in the rat. H is the placebo or just the vehicle injection, and M is the cell therapy. That we can notice that there was no improvement when you inject the saline vehicle, but with time, there was significant restoration of the leak point pressure with the muscle cell therapy and restoring it back to the baseline by about 6 weeks. So it's not right there. It's not an immediate improvement like surgery, but the improvement takes a period of time to achieve, but then there seems to sustain for a longer period of time. And the mechanism of action could involve the cell injection forming new muscle. It fuse and improve the existing muscle, but also growth factor to improve the matrix and nerve innervation. And the technology, this was developed when I was a professor at the University of Pittsburgh and was licensed to Cook, one of the largest medical device company in the world, and I know it's in Colombia, and they've taken it now gradually into phase 3. So I'll give you a little pictures of the snapshot on how the technology has moved forward. I think this was their exhibit at the AUA last year. Initially, now 10 years ago, there's not been a stem cell trial in pretty much any field. So when we applied to the FDA, they were very nervous to approve it. So we tried it in Canada at Toronto, and Senator Hirschhorn, Leslie Carr, urologist at the University of Toronto, and they asked the Health Canada, they're much more flexible. So it took finally 2 years before the US FDA approved the first trial, but in Canada, they approved it in 60 days. And we finished the Toronto trial before we even got approval by the United States. These were 8 women that was waiting to be in the waiting list to have a sling operation. I mean, you heard of the Canada healthcare system. It's very good, but there is delay for elective surgery. So they were all gonna have the sling and they agreed to it. This was the first in human testing concept. Let me just show you then how it's done. So it's the cells is harvested through a lateral thigh biopsy. So the foot would be down here, the head would be up there. It's vastus lateralis. This is the IT band. So it's like a prostate biopsy like needle that you do it from the lateral thigh vastus lateralis muscle. It's done under lidocaine, and we want about 150 milligram. And here's a little bit more picture where the needle, and we want the biopsy to be right there. It's outpatient, they can walk right home. And because we know we need to, it started in Canada, that there's customs, so they had to develop shipping, so we know the biopsy is good for 72 hours before it reaches Pittsburgh, so the trials and future therapy can be done anywhere in the world. And the injection, the cells ship back to the site frozen. Reconstitution is very much like Botox. So the actual treatment part of it, mixing up the cells, just like reconstituting Botox. And for stress incontinence, Cook developed a special injection needle for the woman's urethra. So 4 ml, 125 to 255 million cells can be injected in the mid-urethra at 9 sites through injection needle. After we showed that everything can work, then the US FDA approved it. And the next trial, it was done not only at University of Toronto, but it was at Beaumont and also at Vanderbilt University with Roger Damakowski and Melissa Kaufman. So now we know it's possible. And as most things in the regulatory, you need to do a dose escalation. So we treated a number of women, as you can see the number of patients in each group, at 10 up to 200 million cells in the mid-urethra. So everybody had the same biopsy, but it's a dose escalation trial. And the women has failed behavior therapy, tried drug, surgery, or bulking agent. And this are some in the table, some very characteristic of women with stress incontinence if they join a trial. They're in their 50s, they're a little bit heavy in the US, and the amount of leakage on the bladder diary and on the pad test. And this was really the first multi-center trial. And there was some hint that with time, from 1 month to 12 months, that we see more improvement with the higher dose. Most importantly, we realized the trial is very safe. It's different than a mesh sling or foreign bodies, that there's a little bleeding and pain with the biopsy, but with the injection, there was no retention, no foreign body, no tumor, and none of the usual side effects that bother. So the safety was there for up to over 1 year. The primary endpoint is at 1 year. And at the AUA last year, I think that's in San Francisco, I presented the phase 2-3 data that look at for women. I think the women that's mostly seeking help is women that has incontinence and they had a previous surgery before. So what do we do in somebody that's failed a sling? What's the benefit of another sling? Should they just be relegated to bulking agent injection? So we looked at our trials, the open label study, but also the first phase 3 randomized trial where the autologous cell, 93 patient, was compared with sham placebo injection. This was a double-blind study. The outcome was the diary, pad test, and symptom score. So out of those 2 studies, we realized 38 women has failed previous sling operations, and we want to see how these patients do, because these are the patients we feel is most appropriate and the FDA is most interested for regenerative medicine. They had the biopsy, injection, followed up. For regenerative medicine, the FDA requires the primary outcome to be at 1 year, not at 3 months for overactive bladder drug. And you're required the long-term follow-up to be 2 years. So it's a very long study. And we're looking at these issues. The FDA requires you to look at, well, how many patients had a 50% improvement from baseline? How many was 75%? So this is the baseline demographic. The first column is the open label. And these 2 columns are the randomized clinical trial with cell or placebo. And once again, mid-50s, a little bit heavy women, majority postmenopausal, but not all, half with stress, half with mixed but predominant stress. And you can see they leak quite a bit, more than 4 episodes per day, and they're very bothered. And so we talked about the improvement. 1 year is the required endpoint. And we looked at, they required FDA, who are the patients that had greater than 50% improvement, greater than 75, and who are those that are almost dry, meaning they only leak once, no more than once, a little bit per day. And as you can see, the purple, I don't know what color will come across, but this color here is the open label study. The orange red is from the randomized trial, placebo control, and the blue is the placebo. As you can see, the improvement was greater with both open label and randomized double-blind trial versus placebo. And moreover, as you can see that you see the improvement by 3 to 6 months, but it's persistent by the 1-year time point. And the side effects from the open label and the randomized trial, and there's really no real difference between the adverse events between the cell injected and the saline placebo injected. And there was no serious AE from the treatment. So 8 of the 11 patients is complete in the randomized completed the 2-year. And all of those patients, if they're better by 1 year, they're persistent by 2 years. So the effect doesn't go away like with collagen or other bulking agent injection. So right now, the history has been about 10 years, about over 300 patients been treated, a number of trials been shown to be safe and durable, and it seems to help the sphincter. It's based on the mechanism. So the second phase 3 trials in women with primary and refractory stress incontinence has gotten started, and that trial is ongoing in North America and in Europe. And to switch topic and to finish up, then I want to tell you the recent experience for a condition that's always bothered me in my career because I've never been able to help it, which is the underactive bladder. So as we move forward with injecting the sphincter, it became obvious, can we help to improve the contractility of the bladder so a person can urinate and not need intermittent or indwelling catheters? So the history of underactive bladder, because botanical urocoling doesn't work very well, people try to regenerate the bladder muscle. There's a little bit of history. You're probably aware of it, but just to quickly summarize that in Germany, they talked about using a technique of large muscle myoplasty, where they take the latissimus dorsal, the back muscle, and moving it over from the back to wrapping around the bladder as a plastic surgery, a neurovascular flap. And they treated patient, they show in some patient, they can get them to contract. So you take the very large latissimus muscle, you cut its nerve and artery and vein, you move it in the pelvis, you wrap it around the bladder, and you reconnect it to the pelvic nerve and the iliac vessels to contract it. So it can work in some patient, but the surgery is so big that the treatment is worse than the problem. Why would somebody want to not do intermittent catheterization and to get their entire body rearranged? 10, 15 years ago, there was interest that Tony Atala showed that can we remake the bladder using like a scaffold, to using a collagen base, can you seed a scaffold and then you patch that on top of the bladder? He showed some improvement with 7 patients. That was very exciting in spinal bifida patient. So then it went and moved ahead into phase 3 trial in pediatric centers. And Joseph reported that in 2014. But unfortunately in the trial, that of the 11 patient FDA phase 3 trial, that only a few of the patients has a little bit of improvement in compliance, but it was not significant. The patient were not able to urinate, but 4 of the 11 patients had bowel obstruction and bladder rupture. So the study was stopped and did not move forward. So my passion, this is us now at Beaumont. I came to Beaumont in that there's a history of aging bladder research, but it didn't have a basic lab. So we were able to form a center that focused specifically on the aging failed bladder. We realized we had to develop, let me tell you how a research can start in a medical school. We needed to add basic research lab to our very good clinical department. We believe that there's so little known and misunderstanding about underactive bladder. We had to educate the medical community. And in any research, we have to move it into clinical trial. So we were able to convince our institution to start a new lab. I was able to recruit 4 members, 2 good PhDs, and support people from across the US to feel interested in doing translational bladder research to come join me. I worked with Dr. Ananias Diokno, who's been doing aging bladder research for 30 years, that we put out a pamphlet, books on the underactive bladder to share with the community. And we received an NIH grant on the unmet need for underactive bladder. And now we've had 4 meetings that we are able to bring together the stakeholders, scientists, drug industry, students, physicians, on what underactive bladder means, FDA, on how to move forward in terms with treatment, and clinical trial. So with our experience in the stress, just went to the FDA and asked them, can we go ahead and do the first clinical trial since Bothanicol for UAB? And they approved us for 20 patients at Beaumont. For men and women with underactive bladder, they must have elevated residual urine, and almost all of them are on catheterization. So we start, and the injection technique is very much on the lower right. It's very much like the Botox injection, using the same cell technique. Now we've just finishing up recruiting our last patient. And actually last week, we submitted our first fast break abstracts to the AUA on the results of our first 15 patients that's completed 1-year follow-up. So this is where we stand. It's not over yet, and I don't know if it's going to get approved. I hope so, but this is where we are right now. Cook mouse site is in the early phase of the second phase 3 study for women with stress incontinence. It's called the Celebrate study. It'll be 320 women, multi-center, double-blind, placebo-controlled study. I told you about our underactive bladder, and we're just finishing up the 20-patient physician-initiated study. We will see the results in 2019 and decide about the next phase. But also, and this is something outside of urology, is because of what has happened, that there is a 50-patient multi-center study in Canada and in England for the use of the technology for the anal sphincter for men and women with fecal incontinence. And at the bottom, that there is a group that's interested in tongue dysphagia, that has difficulty with speech and swallowing. They're injecting the autologous muscle cell therapy into the tongue to try to repair the tongue's muscle function. That's in the early phase, but I just think that's just amazing how they can come up with something like that. So this is my team at Beaumont. So and on the top here is my my PhDs. I'm just by pure luck that they are also younger women. My clinical team at Beaumont, Ken Peters, Larry Searle, Diokno, but also the people that's reached out to us with interest in underactive bladder. The scientists that I worked with starting at Pittsburgh, University of Madison's interested working with us with the mice model for underactive bladder, and a number of scientists that we met through the meetings, and that's also is frustrated with the condition. And it's something, obviously, very hopefully, that is something we can add my friend Mauricio Plata to the list in the not too distant future. So in finishing up, this is to me a cartoon of regenerative medicine, that there is a disease dysfunction for us. a muscle dysfunction in the lower urinary tract, bad muscle, can we, using a stem cell from a good muscle, from the patient, him or herself, grow it out and then inject it back into the deficient bladder or sphincter to restore the function and make it a permanent treatment? So I'm just a big fan of Mauricio, and I had the most wonderful time when he invited me down there in the warm weather. He might, I don't know if you remember that the Obama shirt that he gave me, it's a very special shirt for me that I like to wear it once a year and wear it carefully. So thank you for an invitation, and I'm glad I have the chance to share this work with you. Thank you. Is that okay?
That I would like to ask you is, we are using as treated muscle stem cells into the bladder, into the smooth muscle.
Yes.
How does it work? I mean, we have a bunch of students here, and maybe they are thinking about that. They're thinking, how does it work? Because it's different. The muscle you have in your thigh and the muscle you have in your bladder. Why is there an interaction among both of them so it can work? How can be that explanation?
Yeah, and it's a tough question. And we wish there was the same cells with the plasticity stem cell in the bladder, but there isn't. Smooth muscle seems to lack the stem cell source, just like the heart really doesn't have it, so it doesn't regenerate after heart attack. So attempts in the bladder smooth muscle has failed. But skeletal muscle, satellite cells, and we believe our cells is probably one generation, one step earlier than a satellite cell, has tremendous ability to regenerate. I mean, the bladder doesn't heal really well, but the muscle heals. You can get injury, and if you exercise, you can make your muscle 10 times its size. So skeletal muscle for evolution survival is really one of the best, just like bone, to heal and regenerate. So they're by the source. But you are right. How can you put a skeletal muscle into a smooth muscle? And when you put fibroblasts, when you put skeletal muscle myoblasts in the bladder, we've done that earlier on, it's gone within a few days, a few weeks. But when we take our clones of our cells, these are pre-satellite cells. They're close to satellite cells, but probably differentiate into them. And we found out when we injected in the bladder and using the co-localization with reporter genes, with alpha, the smooth actin, we found out there is the plasticity of the skeletal, these cluster of cells, like a stem cell, to differentiate from the skeletal into the smooth muscle lineage. But also there's fusion. These skeletal cells actually fuses with the smooth muscle. So you don't get, in the rat model, we can't do biopsy in human, you don't get sheets of myofibroblasts within the bladder, but you will get these cells long-term with the skeletal cell markers that tends to fuse or sits in isolation. So will they in itself be able to fuse and contract, or do they excrete the trophic factors, neurotrophic factors that would actually help the smooth muscle of the bladder, the innervations of the bladder to improve? Us and other group has demonstrated that there's increased acetylcholine receptors with time after the injection. So the precise answer is we don't know. Definitely you're not getting skeletal muscle in the bladder, but what is the exact interaction? And I listed several things that it can be, but it's impossible to do the biopsies or harvest the bladder in the patient. So everything is an extraction of what we have seen in the rat as possible and potential mechanism why this can't work. So it's an inconclusive answer, but if you look at across specialty, these are things that has been observed and seems to be the consensus on how it can work from one organ to a different organ type of stem cell.
One of the things that is interesting in the randomized control trials is that you exposed patients to placebo.
Yes.
And one of the main concerns we have with the case series is that when you inject something in the bladder or you inject something in the sphincter, it can work because it acts like a bulking agent.
Right.
So it works because you put something in the sphincter. But when you randomize the patients to placebo and you see that the treatments work better than placebo, this is gone. And this is something that's interesting, and we have to wait until we have more information on that. But it's great to see how this whole thing is growing.
You know that too, and you know, even in Botox studies, that I remember that we've all been surprised how few patients, you think it's impossible, but saline injection has helped people with overactive bladder and Botox. So yeah, that's why this is so expensive and it's taken so long. The placebo was tested, so it's reconstituted, and you can't tell. And its power, that you just know the placebo will have about a 40% effect. That's why you require over 300 patients in a surgical trial, and you need 2 years of follow-up. So it's true dedication, but only with this number of patients can you then hopefully see enough separations from the anticipated 40% placebo effect. So knock on wood, hopefully get improved, but people should then be realistic and say, oh, you only beat placebo by 20%. Instead of saying like, that's so little, they should recognize, like you say, that's incredible, because it is so hard to beat placebo, and it's hard to do a long-term study. So that's what I think we're going for. Hopefully there will be, the placebo will be 40, and hopefully the treatment would be statistically significant, but clinically meaningful, more than 20, 30 points greater than the placebo response. But it is hard to do a surgical placebo double-blind trial.
We have around 60 people connected around the world, I think, because there are so many people connected abroad, not only from Colombia. But one of the main concerns in the questions that I'm seeing is cost. It seems to me that there is so much background research that this treatment can be a little bit unaffordable.
Yes.
At the very beginning. Do you have any rough idea about prices when it be commercialized in the future?
Well, the obvious answer is, like any drug company or device company, and I'm not part of Cook, is nobody knows. But I think that just for like benchmarking it with other stuff, is this would be an outpatient, hopefully a single injection and a cure, as opposed to the multiple bulking agent injection, and it can replace the sling. So to me, that anticipation is that it should be, at least in the US, that the reference would be like how much it is to do a sling, not only the material, but the inpatient, the anesthesia costs, because this is very complicated to make. So to me, is like, what is reasonable? Well, it should be, if it's proven to be successful for new and recurrent stress, it should be about like the cost of doing a sling. Should be different than doing a single bulking agent, because nobody ends up with one bulking agent. So it's a very, very rough thing. But how it will play out, the future medicine, and every country is different. And one of these will be one of those things that the cost can improve, because the first 100 patients will be more expensive than the next 5,000 patients. There'll be the technology development. They can make it automated, make robot making the cell. So it's just like the immune therapies. It's a very intensive technology right now, because every patient is the own product. That's what makes it. It's not off the shelf.
I guess the selection of patients was for idiopathic underactive bladder patients, I guess.
Yes.
What are your thoughts about fibrosis in terms of like post radiotherapy, for example? Do you think the cells can grow?
Don't know, but we haven't had it. But definitely, we've had a number of patients. Because we do the trial, we publish. So there's been a number of people with fibrosis, people that's had radiations in the past, and saying, I want to do this for my bladder because there's no treatment for it. So I think it is a very good therapy option. But right now, we want to finish up our underactive bladder and see where we can move forward with that. But you are right. The next urology indication I would love to consider on would be for patient with a fibrotic bladder, especially after radiation therapy.
How much time do you think it will be available for everybody in the world?
Don't know. we need to get one indication approved. It's like it's a 2-year for every patient. So it's like, if you think of 3 months is the outcome for a overactive bladder drug study, each patient is 3 years. So it takes 8 times longer per patient than for any other urinary trial. So it's just a very complicated, expensive process. So, but we started the final women trial, and we had the 2019, the underactive bladder trial would be essentially over, and we'll go back to talk to the FDA.
Now that there's some pressure on slings in the US and abroad.
Oh, yes.
Can you expect that this process can be speeded by the regulatory agencies in the United States as a means to get a new treatment that works better than slings and than the current treatments?
Yeah, you hit the nail right on the head. Slings, any synthetic sling, it's been such a gift to lawyers in the United States, because like many women are having problem after synthetic sling with not only real fistulas or erosions, but pain, dyspareunia, this and that. So it's really very risky to do a synthetic sling. And the FDA has recognized that the guidelines on failed sling, that's synthetic or even autologous, is not that clear, because there's really no strong evidence that a failed sling, nobody really knows what the odds of working on. So we're going to the FDA as we speak, as you speak, saying, listen, this is a high priority. Can you consider this group of women that's failed a previous sling, as being a fast track for autologous muscle-derived cell therapy? Yes, so make it available to them first, and not for faster, before women that just has stress incontinence, that hasn't had a surgery already, because this is the gap right now. We don't know what to do for them, and the cell therapy is safe. So if we prove a degree of efficacy, why not consider, this, it's been now 10 years, hundreds of patients, there's just really it doesn't do anything, it hasn't caused any severe adverse event. Why not consider this as an option for women that's failed a sling surgery? So thank you. That I feel strongly about that.
Okay, so I find this topic a very interesting, a very promising topic. So we are very thankful for you for showing the results, for showing this novel therapy, and we wish for sure that it will be available for everyone shorter.
Well, I hope that this works, that we can get to this step that Ryan and I can visit Mauricio again, because he's the best host.
Great. So thank you very much for your time, and have a great day.
Okay, you too. Take care, Mauricio.
Thank you, Michael.
Bye-bye.